NMR metabolomics. What is it? Today we are going to take a look at this high throughput analytical technique and give an overview of its applications and how it actually works. So without further ado, let's get stuck in. NMR metabolomics. Nuclear magnetic resonance spectroscopy, or NMR spectroscopy for short, is used in NMR metabolomics to analyse small molecules known as metabolites in biological samples, such as tissues, cells, and biofluids. This infers physiological function with respect to metabolic pathways, whereby analysis can be targeted or untargeted. It can be used to determine the pathophysiology of disease, response to medical interventions, and much more, while also providing chemical structure analysis. It is regarded as non-destructive, reproducible, and cost-effective. The NMR pipeline. NMR samples are often stored at -80 degrees Celsius, although depending on the sample, there are guidelines on how to optimize storage and collection to maintain sample integrity. Samples are then thawed out and undergo centrifugation steps and are mixed with a master mix containing chemicals such as azide to stop microbial contamination, TSP to create a known reference peak, and deuterium oxide as a stable isotope of hydrogen that can be used to stabilise the magnetic field strength and reduce the solvent signal. Samples are then placed in the NMR spectrometer, analysed, and NMR spectra is acquired. Spectra then undergoes quality control procedures such as alignment and water suppression, and are annotated with known metabolites and analysed computationally. But how does a spectrometer even obtain such information? NMR spectroscopy is the answer. NMR spectroscopy relies on the physical phenomenon known as nuclear magnetic resonance. This is exhibited by certain atomic nuclei, such as hydrogen. But how does it actually work? Number 1. Samples are placed in an NMR spectrometer. Number 2. Samples are subjected to electromagnetic radiation and a magnetic field. Number 3. Hydrogen atom nuclei present in the chemical structure of metabolites within samples are affected by this. Number 4. Hydrogen starts to exhibit precessional motion at a specific frequency, called the Larmor frequency. For NMR to occur, the electromagnetic frequency samples are subjected to must match the Larmor frequency. The Larmor frequency is unique to each hydrogen nuclei in a compound and varies based on electron shielding, chemical structure, and bonding. Number 5. When resonance occurs, energy is absorbed by the nuclei, going from lower to higher energy states, inducing a voltage detected by the spectrometer. This is ultimately transformed into the NMR spectra output we receive for each sample, which you will see on the next screen. The Skittles analogy. NMR spectroscopy is a complex scientific technique and theory to wrap your head around, involving biology, chemistry, and physics. And sometimes the best way to understand something is to break it down into bite-sized chunks, or Skittle-sized chunks to be precise. Now, imagine you have two different experimental groups, an osteoarthritis group versus a control group, or maybe an original fruits Skittles bag and a crazy sour Skittles bag. We know these two bags of Skittles are likely to be different in some way, but we don't know how many Skittles each bag has or how many types of Skittles each bag has. We don't even know if they have the same Skittles or different Skittles from each other. We might use NMR to find out what each bag contains and how many skittles it contains. We might even substitute the word skittles for metabolites. Now here is an example of an NMR spectra. After running NMR spectroscopy on your samples or skittle bags, you may get an output like this. This is a small section of NMR spectra from a recent study, annotated using softwares and reference databases to determine what peaks correspond to what metabolites, where each peak reflects a hydrogen environment in the composition of specific metabolites, whereby some metabolites may have multiple peaks. When you think of these peaks as reflecting hydrogen environments, it makes sense that the more of a metabolite you have, the more of that specific environment there will be. Therefore, the peaks are likely to be a lot taller. Now, if we come back to our skittle analogy, we can use a key and say certain coloured skittles represent certain metabolites. Now, let's say this example spectra is from our original fruit skittles. We can start to say that the bag has more purple than red skittles, for example. Now, if we repeat what we have done on our crazy sours bag, we could then compare the two spectra to see if they have the same skittles or different ones or different amounts of the same skittles. Applied to metabolomics, this means we can compare experimental groups and start to conclude similarities and differences which might be characteristic of what we are trying to investigate. We can then conduct statistical analysis to determine which are statistically significant and therefore differential between our groups, and we can start to map these metabolites to metabolic pathways, which allows us to infer the functionality and physiology of what we are exploring, which may provide insight into mechanisms that change with disease, for example. Now, finally, a little bit of background surrounding the University of Liverpool's NMR Centre that has supported the production of this video. The NMR Centre for Structural Biology is located in a purpose-built accommodation at the University of Liverpool. The facility has received funding from the MRC Clinical Research Capabilities and Technologies Initiative previously. It houses 3 solution state spectrometers at 800 MHz, 700 MHz and 600 MHz, all equipped with helium-cooled cryogenic probe heads for maximum sensitivity. Here's our building. Thank you very much for listening. This video has been made by Emily-Jayne Clarke of the Institute of Life Course and Medical Sciences and the NMR facility at the University of Liverpool. Please subscribe to our YouTube and follow our Twitter accounts for updates and education resources.